All right, hello, my dears. How are you? Mr. K here. So we're starting a new section. We're actually starting kind of a new part of the course. This is the second half of the course, and it starts with Archaea and Bacteria. We're basically now going to go through the domains and the kingdoms and talk about all the characteristics of all life on Earth. So we're starting with, this week is going to be Archaea and Bacteria and Protista. So we're going to get to the end of that. So all the Archaea and Bacteria notes are already up in Teams, so you can see that. And let's just start. Let's just go over it. So this is your Archaea and Bacteria lecture. It'll be brief for now. And then we'll do the second half tomorrow, and then you're done, Archaea and Bacteria. I'll put a little worksheet, a little review worksheet. So this stuff, Archaea, Bacteria, and Protista, so all the work from this week is going to be quizzable. There's going to be a quiz. It's going to be determined on how far we get to Protista, whether it's on Friday or whether it's on Monday here when you see me in the reel. It might be on Monday in the reel. So study. I will, and there will be a review sheet. So it's somewhere here, and it will be going up on Teams as well. Okay, so let's talk about Archaea. Let's talk about Bacteria. That's Unit F in the book, if you want to have a look. So remember, we already talked a lot about these guys. Remember von Leeuwenhoek? They were first discovered there in the 1600s, late 1600s. Von Leeuwenhoek, a Dutch dude, invented microscope. He was selling drapes, and he needed to inspect drapes, but he got really good at making better and better microscopes. And finally, he saw bacteria, and he was amazed. That's the first time any human in the history, in the millions of years, had seen this amazing microbes. And of course, it opened up medicine and all these things, because people were getting sick. And now finally this led to the discovery that it was these germs, these little microscopic creatures, that were making us sick. And then, you think that all these discoveries and stuff are sort of done like in olden times. But no, in the 20th century, in the 1970s, it started to get noticed. Like when I was little, most people taught that there was bacteria. We didn't know about Archaea, so I'm going to talk about Archaea. I think people knew about Archaea, but not much was known about it. And I remember in the 90s, there was the big news that life had been discovered in these thermal vents deep underwater where no one thought life could live. We haven't really explored that much of the ocean because it's really hard to get down below. And especially in the 20th century, we didn't have the technology to do it. We do now. I mean, James Cameron went to I don't know, you guys know who he is. He's like a movie director. He went down to the bottom of the ocean. But they discovered these methanogens, these bacteria that can live next to these hot thermal vents. When I was a kid, what was taught was that only sunlight is the only thing. All life begins with sunlight. Without sunlight, you couldn't have any life. But at the bottom of the ocean, there is no sunlight. So how do these things work? Well, they take the chemistry and they convert the inorganic chemistry, the inorganic molecules, into organic molecules that they can then metabolize for energy. It's not the most efficient way to do it. They're known as Archaea because they're considered the first bacteria. They're ancient. Archaea means archaic, means ancient, really old. So the big difference between Archaea, and they were only discovered in the 70s, and they haven't been studied that much because they're really hard to study because they live in horrible conditions. So in the lab, how do you reproduce those horrible conditions without hurting yourself? They live in places where we absolutely could not live, hot springs at Yellowstone National Park, and they sulfur lakes and salt lakes. And the reason they can survive in these places is because of their cell membrane. Remember the lipid bilayer that we learned about? So regular bacteria, us, we have a lipid bilayer. And so it's those phospholipids that looks like this. They've got the hydrophilic head and the hydrophobic tails, so these don't like water, and they arrange themselves, remember, in that lipid bilayer. Well, Archaea have a lipid monolayer. These things are actually one chain. They're super hard, and they have different bonds here. They have hard bonds, and so it makes a much stronger cell membrane that can resist temperature, can resist heat, can resist acid and all those things. So they actually have a mono layer, but it's got hydrophilic ends on both sides. And then they have proteins that let stuff through. What else do I want to say about Archaea? They're prokaryotes. All of these bacteria we're going to be talking about right now are prokaryotes. So you remember prokaryotes have no MBOs, no membrane-bound organelles. No membrane-bound organelles. But they do have some things. Here's a bacterium, and they've got a nucleolus, or nucleoid, sorry, not nucleolus, nucleoid. That's just the lump of DNA. It doesn't have a membrane or anything around it. It's just a lump of DNA. They've got ribosomes. Remember, there was a question on some test. Ribosomes, if you don't have ribosomes, you can't synthesize protein. So they've got ribosomes spread throughout the cytoplasm. They got plasmids. These are little bits of DNA that they can share between other bacteria. They're like genes, basically, a little thing of gene. They're very dangerous if these are pathogenic bacteria. They have these things on. So they have a cell wall that's made of sugar and protein. So that's the cell wall, and they have a cell membrane, just a regular cell membrane. Or if they're Archaea, one of the special non-bilayer cell membranes. They have obviously a cytoplasm. So all the energy production, everything just happens in the cytoplasm. There's no mitochondria, there's no things. It just happens across this membrane. It's not that well organized. Remember, we talked about that. And some bacteria have a flagellum, and some bacteria have these things called pili. There's these little hairs, and what the hairs let them do is just attach themselves to places. They're like a cush ball. Do you guys remember? No, you don't remember. Those were like Velcro. Sure, like Velcro. It can attach itself to things. It's got little hairs. So that's kind of that's the structure of bacteria. Oh, I've messed up my. There we go. What else do I want to talk about? Some of these Archaea have been found, and that structure of the bacteria applies to both Archaea and bacteria. It's just the cell membrane that's different, but all the other stuff they basically have very similar. And people used to think that Archaea were bacteria. So we didn't have domains before when Mr. K went to school. And when he went to uni, there was no domains, or domains were just sort of like, I think, being, are we going to do domains or not? So it just started with the kingdoms. And bacteria and Archaea were in the kingdom Monera. So you might hear about Monera, and Monera included Archaea and bacteria. And at the time, bacteria were called eubacteria. So now they're just known as bacteria. Remember, when we learned about taxonomy, people make it up. They make up the classifications based on criteria, and the more we learn, the criteria change, the names change. Now, some of these Archaea, one place where they live, where they've been discovered, is they live in mammal stomachs. So not us, we don't have Archaea living in us, but for example, cows have these bacteria, and they are methanogens. So they metabolize. Their part of their metabolism produces methane gases. So cows fart a lot of methane. It's a greenhouse gas. So it's a bit problematic. But what the methanogens do, it's a symbiotic relationship, which means that the bacteria benefit and the cow benefits, because what they're able to do, those bacteria, is help digest food that the cow can't digest. So these methanogens, they have the enzymes to break this food down, which is grass, I guess. Does that cow eat grass? I'm not a farmer. So it's probably those cellulose things that are really hard to break down. Okay, I think that's enough. So we talked about Archaea. Have a look through the notes. You know, today, if you can get to let's look at the structure. Tomorrow, I'll tell you about gram staining, and then there's quite a bit of stuff here for 2 days, but we got to do it in 2 days, and I can't make these videos too long because you guys won't watch them. So read through this, look at all of this thing, and I'll go through some of the tough stuff, more tougher stuff tomorrow, and I'll put a worksheet up, and there will be a quiz on this most Friday or Monday, to be determined as we see what's going on, okay? Yeah, all good. There we go.